I'm John Northrup, and my passion is to bring better therapies to cancer patients. We are doing that with Stingray Therapeutics, bringing forward the next revolution in immune oncology. We're raising a $2.5 million preferred seed series 3, with half of it already taken up to complete our phase 1 clinicals. And I hope you might join me in this incredible effort. Now, to explain just a little bit about what we're doing, today's solid tumor immunotherapy cancer drugs are checkpoint inhibitors. They work in only one arm of the two-armed immune system we have, adaptive immunity. This is T cells to fight the specific cancer in the body. The T cells enter the tumor and tear it apart from the inside. But cancers are very smart. They make large amounts of so-called checkpoints. Think of these as border crossings, refusing to let T cells cross into the tumor. So checkpoint inhibitors take down these checkpoints, and T cells can get into the tumor and fight again. Unfortunately, these inhibitors only work in about half of all cancers. Why? Because our immune system is two-fisted. The other key arm is innate immunity. When it does not function, checkpoint inhibitors cannot work. Turning on innate immunity after cancer has suppressed it is what is needed to take the next big step in immune oncology. Cancers suppress innate immunity by making a lot of a protein called ENPP1. It is the only known innate immune checkpoint. Our drug blocks ENPP1 and lowers this checkpoint, so innate immunity joins adaptive immunity in the fight, and T cells can rip into more tumors and tear them apart. By using our ENPP1 inhibitor with checkpoint inhibitors, we can make immune cancer drugs work in most of the other half of all tumors. Now, I'd like to show you just a little bit of scientific data. So here we're flatlining this colon cancer in a mouse by using our drug with checkpoint inhibitor. This is just one of the many proofs of concept we have done. We're also very synergistic with radiation. Radiation excites innate immunity, and we have great synergy with the many new targeted radiation therapies coming to cancer in the next few years, with 2 already on the market. Now, this shows how in cancer, high ENPP1 means very low checkpoint inhibitor response. Checkpoint inhibitor response rate is on the y-axis. These are all very low responses, as this is 25% to 0. And a good response for checkpoint inhibitors in melanoma, and it's 85%. And on the x-axis, we have the log of ENPP1 expression in these cancers, going all the way to a log of 12 or a billion overexpression. So this shows that cancers have an enormous chance to build ENPP1, and by doing that, suppress our ability to treat them with immune oncology. And we have an enormous chance to work miracles in these cancers that have high ENPP1 expression. And we have picked the 3 bolded cancers to start in. Now, this is a schematic of the clinical trial plan that we have, and we are filing our IND, permission to do clinical trials, literally in days, and we expect to start these studies this summer. The first one would be ENPP1 plus radiation in pancreatic and colorectal cancers. And then we would go on to renal cell carcinoma and hepatocellular carcinoma, plus double checkpoint inhibitor. And then in microsatellite stable colon cancer with 1 checkpoint inhibitor. We have 3 completely chemically distinct clinical scaffolds. Scaffold 1 is our lead candidate. They're fully owned by Stingray. We have no economic obligations to any university or anyone else. And we have 4 patents received and 3 patents in process. Now, this is the competitive landscape, and like with any truly exciting program, there is competition. But we are neck and neck with Riboscience, who just started their phase 1 clinical last year. We're the next program up, well ahead of the others. And by the way, our compound and our clinical strategy is way better than theirs. This is our time to exit and the capital we need. In total, we expect to raise this seed series 3, and then a second raise after phase 1 for 10 million to do the phase 2. That's all the raising we plan to do, and then exit in 2025. Now, these are buyers and sellers of programs in our space, the same exit we planned for. And this is just a straight average of recent exits directly in our space and our precedents for the deal we're after. Returns here have been incredibly lucrative. And it's probably because the potential is so great in terms of the ability to extend immune oncology into far more tumors. We're specialists in biotech and pharma, now bringing you our second company, with the first doing very well on its own. We can execute and know the space. Our team is strong. Scientific juggernaut Sunil Sharma, my co-founder, has done over 150 phase 1/2 clinical oncology trials for all the major cancer companies and biotechs as primary investigator. I had 28 years in big pharma at Eli Lilly, with the last 14 years as SVP in corporate business development, where I did 40 deals like Stingray on the buy side. I know business development people in all the cancer pharma companies and how to talk to them. I've been CEO of several biotechs over the last 15 years, and we have built this team together across them, and they're all fantastic, experienced experts. This is a security we're raising. It's a $2.5 million preferred seed series 3 at a 17.5 million pre-money. Half the raise is already funded, with 1.25 million left. Biotech can be very lucrative. If we exit on time and even spend another 5 million more than we plan, an investor in this round will see a $4 share price return, and potentially $100 a share in milestones and $750 a share in royalties across the product's life. And we're built to be sold. We have a lot of traction. Preclinical is done. We're close to exit with a big potential drug, and we have some notable investors in our program. Our raise is open now and will close soon. Join us. Talk to me further, and I'd be happy to take the deep dive with you on Stingray Therapeutics. Thank you very much.